Genomics, Transcriptomics, and Proteomics, presented by Macy McHouston and Hannah Morris. Structural genomics is the study of the organization or sequence of genetic information. Genetic maps is the map of relative distances between chromosome regions. They are measured in recombination frequencies or map units, and they are based on crossing over rates. Physical maps are a map of physical distances between chromosome regions. They are often measured in base pairs, and they are based on direct analysis of DNA. They are also more accurate than genetic maps because they are higher resolution. Genome sequencing is used to determine ordered nucleotide sequences of organisms' genome. Map-based sequencing involves short sequence fragments that are assembled into a whole genome sequence through chromosomes being separated, and then each chromosome is cut up by partial digestion, and then they are fragmented into smaller clones that are used for sequencing. Shotgun sequencing involves small insert clones that are prepared directly from DNA and then sequenced. And the DNA is cut into small overlapping fragments, and then those fragments are sequenced, and the overlaps are used to sequence those clones, and then the computers assemble the entire sequence. To make sense of DNA, we can often use bioinformatics, which combines molecular biology and computer science to use statistical analysis to solve biological problems. We also use metagenomics, which is where we determine genome sequences of groups of organisms that inhabit the same environment. And finally, synthetic biology is used to design new organisms that hopefully provide useful functions, such as microbes that provide clean energy and break down toxic waste.
Transcriptomics is the examination of the expression of the genome. One method is to use microarrays. Microarrays rely on nucleic acid hybridization and use known DNA fragments as probes. These fragments are affixed to a solid structure in a series of dots referred to as an array. They are marked with fluorescent markers and will fluoresce when complementary strands hybridize with them. Another method is RNA sequencing. It can be done in various ways but includes the following: isolation of RNA molecules of interest from the cell, conversion of RNA to complementary DNA using reverse transcriptase, fragmentation and preparation for sequencing, sequencing of the DNA amplified via polymerase chain reaction, and assembly of the sequence reads into RNA transcripts. Proteomics is the study of the proteome, a record of the cell's protein content. Methods used to determine cellular proteins are two-dimensional polyacrylamide gel electrophoresis, also known as 2D PAGE, liquid chromatography, or mass spectrometry using traditional or shotgun proteomics. In order to study the protein structure, two methods could be used: X-ray crystallography, where the crystals of the protein are bombarded with X-rays and the diffraction patterns studied, or nuclear magnetic resonance, shortened to NMR, which provides information on the positions of the atoms within a molecule based on their nuclei's magnetic properties. Affinity capture uses an antibody to a specific protein to capture a protein from a complex mixture of proteins. The captured protein pulls down proteins it interacts with. The captured mixture is then analyzed via mass spectrum. Protein microarrays are similar to DNA microarrays. Proteins are applied to a solid support in an orderly array of dots and marked with a fluorescent marker. A spot will fluoresce when the protein bonds to an antibody, indicating the presence of the corresponding protein in the tissue sample.